Hi, I'm Barbara Tharp.
And I'm Deanne Erdman. We're from Baylor College of Medicine Center for Educational Outreach.
Today we're going to look at observing different microbes. It's the fourth lesson in the Science of Microbes series.
And you'll find that all of our activities are aligned with the National Science Education Standards, and this goes along with both inquiry and life science standards.
In this activity, students will use a microscope to become familiar with 3 different microbes: bacteria, fungi, and protists.
All right, great. So we always want to, when we're working in groups, you have to have a lot of classroom management, and we think it's important for you to spend time with this procedure in the beginning of the year, and then as you go along, the kids become very practiced at working in groups. But my best advice is to plan simple activities the first time you do something with your students, and to be methodical. Don't be in a hurry. If it takes you 3 days to get the activity done properly, take 3 days, because once the students learn their roles, and there's lots of suggestions in the guide, such as one student can be a reporter, one can be a
materials manager, another one the maintenance director, and another one the principal investigator. And as Deanne says, once they understand what their role is, then you have the management pretty well under control. And when I talk about the different roles, they will rotate through the roles. They won't always have the same role, but they'll understand all the different roles. And it's important that you understand that, for instance, if you're the principal investigator, that doesn't mean that you're the only one that gets to touch anything. Right. It just means that you're in charge of making sure that everybody does get to do the exploration. And if you're the maintenance director, it means that you make sure things are cleaned up. It doesn't mean that you have to clean up everything. But the roles will rotate, and the kids after a while become very comfortable with understanding. They come in, they know their groups, they know the procedure, and then it takes science to a new level, I think, really.
Well, and plus that's something we hear from industry all the time about students and people out in the workforce in general, is that very seldom do we work in isolation. Exactly. And so learning to work in a group is such a good skill. So in this particular activity, you're going to need to plan ahead. You need to get some pond water. There's lots of sources for that. Sometimes I've had students bring it in, but you can collect it from drainage ditches, around golf courses, a pond, and or there's sort of a recipe for creating a pond water using hay or grass in the guide if you just don't feel like you have a good source at all. But you do want to plan ahead. If it's, say, a cold time of year, you don't want to go out and get your pond water the day before you're going to use it, because you're going to have some disappointed kids when they don't see very much activity in that pond water. So bring it in ahead of time, let it warm up, let it grow.
And also you can order it if you must.
Yeah, absolutely. Then, in addition, you're going to need some yeast, just normal baker's yeast. Mm-hmm. And we suggest that you mix that up with a teaspoon of sugar, and you do that overnight so that you'll have a lot of good growth in it. And then you're going to want some yogurt as well. And once again, with the yogurt, we just put a small amount in a small container for the students, and we left this out overnight also. So we're not.
It's not to eat, but it looks a lot more exciting after the microorganisms have had a little bit more time to I guess rummage around in there.
Yeah, absolutely. Then we have some pipettes. We have some clean blank slides. We have some thin, small cover slips. We have toothpicks. Sometimes when you're working with microorganisms, you can order this from any biological supply house, and this is glycerin, and it helps to slow the little microbes that are swimming around in the pond water down. And then you may need something to measure with as you're preparing some of the original solutions. All right. Safety. Always want to pay attention to safety. It should be a practiced part that students are always expecting to write down something, read something, talk about safety before every lesson. Even if you have these lessons, these rules posted in your classroom, don't assume that students, because they've been in your room half a year, don't need to hear safety again. Always talk about safety. Follow your district and school guidelines. Have students read and follow instructions. Even something as simple as spilling water on the floor. If a student doesn't report that, somebody could be running and slip and fall, and it could cause a bad accident.
And also the tasting part. That's always important, especially with anything that might have food involved. And even though this is not food for consumption, just to reiterate the fact that this is not to be tasted.
Absolutely. And we always want to wash our hands when we're finished. All right, so to kind of focus in on what we're doing today, we want to kind of start out with some questions.
Yeah, I think, for instance, you could ask the students, can you name a microbe or a microorganism? A lot of them probably might come up with just like bacteria or a virus, for instance, and you can just record those basically, and say you're gonna revisit.
Absolutely, and if students are completely off target, you want to maybe kind of redirect them. But the idea, and it's certainly not anything wrong with saying yes, that's absolutely correct. But we don't want at this point to get into a sort of stand and deliver kind of teaching style where you're now telling them everything that they ever didn't want to know about a microbe. Instead, we want them to kind of be excited about the process of discovery. But we want them to kind of think about maybe how narrow their vision is of microbes.
And there are a lot of questions that you can ask, and there are a lot in the teacher's guide just to get them thinking about questions. What do they know about them? What are they wondering about them? Do they know what sizes they are? A lot of different things that just to start getting them excited about where they're going with that. For instance, do all microbes look alike? They don't know. Are they going to find microbes? If we look at the next step, which is we're going to look at yogurt, and we're going to look at yeast, and we're going to look at pond water, are we going to find microbes there? That would be a good question to ask. I think that there would be a lot of kids that would be real surprised with what they're going to find.
I think so too, and I think that you'll probably find that most students have the misconception that most microbes are harmful, and just the opposite is true. All right, so we've got some student instruction cards for students to be able to follow along, and once again, as we are working with students, we put them in groups of 4. And you could approach this several different ways depending on the number of microscopes you have, depending on the amount of time you have, depending on the skill level of students that you have. Sometimes you have a real mixed skill level in your classroom. So you could actually let students make all 3 slides in their group and look at them, and that's going to be more time consuming, but certainly they're going to have a lot of fun doing that.
Or they could work in teams, and you could have 2 teams that would actually do the paramecium, 2 teams that would do the yeast, and 2 teams that would do the bacteria. And once the slides were created and the observations were made, and they were focused like they need to be, so they could really make good observations, then they could go around the room and they could look at each other's. That's another way to do it. Make it a little bit quicker and less complicated.
And another thing that I've found that's useful sometimes is you can periodically send an original member of the group back to be a teacher, where they're explaining to the new group of students rotating through exactly what they're seeing and why. So we always know that once you can explain something, you really do know
exactly.
it
That's
a
great.
lot
That's a
better.
great idea.
Okay. All right. So we're going to make some slides, and we've got some student information cards that are going to guide you through this, but I'm going to just kind of quickly sort of talk you through it. The first one that we're going to be looking at is bacteria.
Yes. We're going to do the yogurt first, and the yogurt is the bacteria. So we're going to take just a little bit with a toothpick. I think would be a good way to do it. And we have all the directions on our card, but basically you get a little bit of yogurt, you put it on there on the slide. Okay. Use a pipette or dropper to add 1 drop of water to the yogurt. And I'd say be careful with just a little drop. Then you're going to use your toothpick to stir it up. I can maybe stir it up.
Okay.
So we're stirring up the yogurt. We have a nice little mixture now.
Right.
Okay. Okay, then again, using the Deanne technique, put your little cover slide on very carefully.
Okay.
At an angle and let it kind of fall down there carefully. And you have your yogurt slide ready to go.
And I have a slide ready to go. And so we're going to put this on the microscope. And with that, we'll start out focusing it on low power so that we can see the biggest area to begin with. And then we'll move up to the middle magnification, which on most microscopes means that we're magnifying about 100 times. And then with this, because the organisms that we're looking at are very small, we're going to need to probably move up to as high as the microscope will magnify, which would be 400 on the one that we've been using.
And what do you think they're going to see when they get to that point?
Well, we're going to need to increase our magnification pretty high on this one because the bacteria that are in yogurt are really very small. They're rod-shaped, and many times they will string together. They do move around. They do have flagella, but certainly we're not going to see the flagella even if we're able to magnify it at 400. But you'll be able to see a little bit of movement and sort of almost like little tiny dashes moving around in the yogurt. And if you've left it out overnight and you've got a good active culture, you'll see a lot of that movement.
And so they're one of the 3 basic shapes of bacteria, which are the rod-shaped, right?
Yes, they are.
And what are the other shapes?
The other shapes are sphere-shaped and spiral-shaped.
Wow, that's cool.
So we have the 3 basic types of bacteria, and they're all considered microbes.
All right, so that's our first slide that we've created and explained. So let's go on to our second, which was the yeast. With the yeast, it says, following the directions, it says, using a pipette or dropper, place a small amount of yeast mixture in the center of the slide. And again, it's much better to let it sit for at least a day before you do this.
All right.
And this is so simple. Now all we have to do is the cover slip. Okay?
Oh, that's great. All right, so I'll get my cover slip, and they can kind of disappear on the paper towel. You have to be careful. And I'm going to once again carefully lay it on there. And now I have a nice even smear of a yeast mixture.
Okay, and this time, is it going to look just like the bacteria?
No, this is a whole new organism.
Wow.
And in fact, it's in a whole different group. And in this particular organism are much more rounded, and they actually reproduced by putting a little small bud off, and the bud grows and grows until it finally pinches off. And that's how they reproduce asexually. So once again, when we put this on the microscope, the students are going to want to start out at the low magnification in order to get focused, and then we're going to move to the middle and then on to high magnification again. And remember that these are not stained, and actually the staining process for working with bacteria, working with yeast, is fairly complex. It's much more than just adding a drop of iodine or adding a drop of coloring. There's a whole special process you have to go through in order to get it to take into the cell. And so that's why we haven't stained them, so that we can see them more clearly. But once again, when the students are looking in the microscope, they need to just settle down and watch it for a few minutes and think about the shape and think about what they're looking for, and they'll be able to see some of those yeast with the small buds coming off.
Okay, and again, we make sure that after they have observed them, that they draw them to the best of their ability.
Yes, absolutely. We want to put as much detail in as they can. Now, they're not going to have a tremendous amount of detail on looking at the bacteria and looking at the yeast because they are, really quite small.
But just the basic shapes.
Just the basic shape.
Right. And they will be different. And then we have a third slide that we're going to create, and that's the Paramecia slide.
Right. And this particular container, I also have a little bit of an aquarium plant in there. But we were lucky in that we have a science center near us in the Houston Independent School District, and we can easily get cultures of Paramecia. And so, and you can order these from Biological Supply House too if you don't have that kind of a science center in your district, and they're very easy to maintain. But I do recommend, if you want to see a large number of Paramecium, going ahead and ordering them. If you get ordinary pond water, most of the time you can find a number of Paramecium in there, but Paramecia. But to make sure you have a large colony, you may want to order them.
This time I'm going to put a nice drop in the center. Okay. Oh, it's a beautiful drop. Okay.
It's a beautiful drop. You want to try the cover slip?
Yeah. Cool.
There you go. Cool.
Okay, now mine's invisible almost, but when you look at, it's under there.
It's under there.
Little bitty moving around little animals. It's so cool.
Little animals. And so again, or not really actually animals, but little
animalcules.
Animalcules. Yeah, I think so. Now we know how they got that name. When we're looking at some differences between all of these organisms, the first thing that we looked at, we talked a little bit about, was the bacteria in the yogurt, and we said that they were rod-shaped. and that the other two shapes are spherical and are a spiral shape. But these are very, very small. They're between 1 and 5 microns. Now, you know, I've been looking through the microscope for a long time, and it's hard for me to even kind of get my head around, you know, what is 1 micron? How big is that? And so later on in this unit, we're going to create a model where we can kind of get an idea of how big that really is.
And the way we're going to do it is we take a Helvetica period. I think it's a 12 font Helvetica period, and I blow it up to 2.5 centimeters square. So that little bitty period at the end of the sentence becomes a 2.5 meter square. And then we place the different microorganisms on that in at scale size. And when we look at 5 microns, it ends up being almost just a little bit bigger than a dot on the paper. So we're talking extremely small microorganisms.
And so that's a great way to talk to students about, you know, why we had to wait for the development of technology to even be able to study and understand some of these kinds of things. It's very difficult, of course, to see the internal components. And when we're looking at bacteria, this may not be something that you want to go into with your students, or depending on their level, but bacteria are what we call prokaryotes. And prokaryotes mean that they have an unorganized nucleus. They do not have a nuclear membrane around their hereditary material. They do have the genetic material, but it's not organized into a membrane-enclosed nucleus, and that's what a prokaryote cell is, and they're much more simplistic, and the bacteria are in that group. Then the next thing we looked at were the yeast, and they were more or less spherical in shape, and they reproduce by budding. And about how big are these, Barbara?
They're only about 10 microns.
Oh, okay. So they're quite a bit bigger than the bacteria.
Double the size, so you're a little bit more likely to see those, but still they're exceedingly tiny.
And, you know, you may decide that you want to give that information to students up front when they're making some of their comparisons, so that they understand that you know, when they're trying to get an idea, are they seeing the yeast, are they seeing the bacteria, and kind of in comparison with one another, because that's you know, one of the skills we want to develop. All of the rest of the organisms that we're looking at outside of the group of bacteria all are a type of cell called a eukaryote, and a eukaryote has a membrane enclosed nucleus, so it's a much more complex type of cell. It's interesting that the way that yeast, this baker's yeast, is used. When the yeast, we feed it a little bit of sugar, and it starts to reproduce and grow, and it's using oxygen, and just like our cells, it gives off carbon dioxide. Only a lot of that carbon dioxide gets trapped in the bread dough, and that's what causes it to rise. I don't know if you've ever been around bread dough when it was rising. If you go in and you poke your finger at it, it causes it to kind of deflate.
You have to be very careful with it when you're in the oven.
Yeah, absolutely. And if you've ever had a chance to go to San Francisco, in addition to the yeast, they actually add a little bit of a particular species of bacteria that adds an additional flavor. Sourdough bread, absolutely. There's another species of yeast that's used in the processing of wine and alcohol, and what happens with it is it gets into a condition where it doesn't have much oxygen present. And as soon as the oxygen is depleted, it goes into what we call anaerobic respiration or fermentation, and it produces ethyl alcohol. So it's kind of interesting, and right away we can see that, you know, we've just looked at 2 microorganisms here, and we see that they're not harmful at all, but instead actually beneficial to the food industry.
And so what would we be without bread and wine?
Absolutely. The next thing that we looked at were the Paramecia, and they're in the Protus group. And, you know, when you look at that shape, Barbara, what would you describe it as?
Kind of like a little house shoe or slipper shape.
Kind of a slipper shape. That's very traditional. And there's a number of different species of Paramecia. They have the long, sort of curved access on the side. That's the oral groove, and that's actually where they sweep food into the organism. there's some simple feeding experiments that you can do if you wanted to do this as an extension with your class, where you actually feed them little pieces of food that has a colored dye in it, and you can see that being incorporated into the Paramecia.
Wow.
So it's kind of fun for the kids to do that. Now these are we've almost got a fish story going here. This is 250 microns, so compared to our poor little,
it's huge
bacteria
huge.
That we were looking at. So you can see that we're already arranging quite the gamut in size, but we're still pretty small.
Definitely.
And again, the type of cells are eukaryotic cells. So, kind of for an extension, something that I think is a lot of fun, this is a great time to actually look at some pond water. And we talked about bringing that in the beginning, and letting students just make a number of different slides and look around to see what kind of microorganisms that they could find in there. Now, actually, they're going to find things other than just microorganisms. They're going to find larvae of multicelled invertebrates like Daphnia.
Mosquito
larvae are very exciting. They'll actually even have like a little siphon tube.
They look like monsters.
They really do. And they look so large compared to everything else. But the students will be able to see a number of organisms. If you take a look at the slide, you can see the hydra, which actually it is another multicelled organism. It's in the family with or in the group with jellyfish. But you will be able to see a number of other species of Paramecium, little one-celled organisms that have little fine cilia around them called ciliates. You'll see a number of different algae. There's an example of Spirogyra on the screen. And some of them will move, some of them don't move. You'll have a difference in color. Some will have chloroplasts, some won't. One of the things with the power of the web, you can find just beautiful websites now that have a number of these that will help you and your students to identify them. Don't feel intimidated if you can't identify everything in that pond water. I don't really know that anybody can identify everything.
But it's like a whole foreign terrain, and it's extremely interesting for students. It really grabs their imagination. It's almost like going to outer space to see all the different cool things that can be in a teaspoon of pond water, or a teaspoon that came from the ditch. Just so many beautiful different kinds of organisms. So this is something that you don't want to miss.
Absolutely. And then don't forget, pull that concept map out and add on some more information, and keep modifying and pushing the students in their quest for learning exactly what microbes are.
Okay, thank you.
Thank you.